in the presence of indole pyruvate decarboxylase to become indole acetaldehyde. Now the again point is that finally the indole acetaldehyde oxidized. It is now see first reaction is the transamination, second it is the decarboxylation, and the third it is the oxidation. Means three phase reaction is that first finally indole acetaldehyde oxidizes to become indole 3 acetic acid, to become indole 3 acetic acid. So this reaction is catalyzed by enzyme indole acetaldehyde dehydrogenase. Means three step reaction from tryptophan to indole acetic acid. There is a three step reaction: transamination, decarboxylation, and oxidation. So these are the three step reaction. That three step reactions are being catalyzed in the presence of three types of different enzymes: transaminases, then decarboxylases, and the dehydrogenases. है ना? So these are the three step reaction in the biosynthesis of indole acetic acid. Example: when we take pea shoots, cucumber seedlings, in that case the tryptophan occurs sporadically, means it is a spread. It is in a spreaded form in all the cells. That's why it is called as a sporadically in higher plants. So it was firstly isolated from acacia. Now it is found in the different several species. C. The second, the tryptophan decarboxylase. See, the reaction is that from the tryptophan there is a formation of indole 3 pyruvic acid, and indole 3 pyruvic acid there is a decarboxylation, indole 3 acetaldehyde, there is oxidation reaction and formation of indole acetic acid. So these are the pathways. Then in case of these different pathways, the deamination, decarboxylation, so these are the stages in the formation of. This is the one single pathway. There are three another different pathways for the biosynthesis of indole acetic acid with the help of tryptophan amino acid. In the next slide we will see. Now what is the physiological role of auxin? What is the physiological role of auxin? Means in the growth of the plant, what is the exact physiological role in case of the cell elongation and longitudinal growth? The first of all, the first effect or the first role, that is in the cell elongation and in the longitudinal growth. Now see, in the diagrammatic representation you will see the growth of the cells in length, it is called cell elongation. That is the simple definition. The primary and chief function of auxin in plants is to stimulate cell elongation. The extensive studies on the mechanism of action in cellular elongation shows that auxin promotes cell elongation by causing increased wall plasticity with the decreased elasticity. Means when now what is the exact role? Means the role of auxin is that the particular auxin components have the capability to stimulate cell elongation, and these cell elongation took place by the auxin by increasing the plasticity in the cell wall, so that gives the elasticity, and decrease the elasticity means there will be a plasticity. If there will be elasticity, what happen? Cell elongates and afterwards it also goes in a backward manner. So that is nothing the action of auxin. That is not the role of auxin. Auxin role means gives the only length to the particular cell wall or the cell to elongation. And after that, and these are been due to the enhancing the water and solute uptake. Means what happen when the solute uptake and the water uptake, there is a interaction at gene level by synthesizing enzyme that require for the synthesis of cell wall and cytoplasmic components. Means when the cell elongates, at that time there is a gene level synthesizing enzymes. So at that time they will perform their role in the formation of cell wall and the cytoplasmic components. That is when the cell elongates, so at that time all the components have also been simultaneously synthesized. Why? Because they have to maintain the elongation of the cell wall. If they does not, if they, it is not going to took place, the cell will shrink. So that's why the cellular components, cell wall or the cytoplasmic components are being synthesized. So auxin also induce enzymatic and non-enzymatic deformation or loosening of cell wall by breaking the cross link between the cell components. Means also auxin also induce, see here in the diagram you can see the what happened, and these auxin causes cell elongation in the shaded side. Means see, that is the stem part, the light comes here. At that time the auxin stimulates the elongation in the cell wall where the shaded side, means here the sunlight comes, and at that time here is the second part is the shaded part. So that shaded part will move because the elongation took place at that time. So elongated cells are in the shaded portion. So see the shaded slide of shoot, and auxin causes cell elongation in the, because the light is coming. So that is the actual role of cell elongation in the of the auxin in the cell elongation. The second role of auxin is that is the apical dominance. What is apical dominance? Is that when the apical bud had been cut, what happened? What is the apical dominance? See, the most of the auxin is formed in the apical region, and when the particular part has been cut, so auxin has been transferred to the basipetal succession through the basipetal succession, and when it transferred to the basipetal succession, when auxin comes in the nodal region, at that time due to that auxin, that is a concentration of auxin increases here, and at that time there will be a lateral movement. Means that particular part has dominancity to transfer that particular auxin molecule, and this goes to the lateral development. So, and this was expressed by Thiemann and Skoog. So this apical dominancity concept was put forth by Thiemann and Skoog in 1934. According to them, the auxin is synthesized in the apical meristem, from where it is translocated to the downwards, causing inhibition of lateral buds. During downward movement, auxin, some correlate inhibitors, which synthesize the growth of the lateral buds. Not the lateral sides are being, the lateral buds are being synthesized. So that is the exact role of auxin in the case of the apical dominancity. See here. Now the third part is that is the cell division. In the cell division process, see the diagram that you have in your left side, the diagram you will see, the first diagram here, the auxin initiates the cell division in the tissues like cambium. So the cambium region is very much capable to, or the cells in the cambium region are very much capable to promote the cell division by the auxin action. So the effect of auxin is particularly important in secondary growth of the stem and in the differentiation of xylem and phloem. Means in case of auxin, the particular effect of auxin is in the cambium region, cork cambium region, in the formation of secondary growth, and there will be also a differentiation of xylem and phloem. So that is the exact effect of auxin in the cell division at the time of in the cambium at the time of secondary development. See here, I am zooming the image. In that you can see the cambium region, the D region that you are going to see, the cells are been, the multiple cells are been formed in the cambium region. So this is nothing but the D region that you are going, and the E region, the consisting most, so auxin has been transferred to the division there is a cell division has been took place for the secondary development, है ना. Now we will see the another role of physiological is that it is nothing but it is the root growth and its initiation. So in the same concentration of oxygen does not show the effect on the different plants, means different plants having not the same concentration, they require 10 ppm concentration for shoot, है ना. In case of you can take the example of avena, but in the different plants there are different concentration required, but the optimum concentration is a 10 ppm. So but in the different plants that have the it may vary according to their growth pattern, it may vary according to its growth pattern. Afterwards, sometimes concentration of oxygen which increases growth of organ inhibits the growth of other organ, है ना. In case of root, the number of lateral branches of roots in increased. So the in case of you will see the quiescent center consisting, that is the in case of the different root growth, there is also a formation of in the root growth initiation. So the exact effect of oxygen is that the concentration of oxygen which increases growth of one organ, and in case of roots it reduces the growth of roots, but the lateral branches of the roots are been growing increased condition. So the main root is been stopped and the number of multiple branches are been lateral branches are been synthesized by the action of in case of oxygen. The fifth physiological role of oxygen is that in case of parthenocarpy. Now what is parthenocarpy? Parthenocarpy, it is nothing but it is the formation of seedless fruit without fertilization. It is called parthenocarpy, although it is a common nature in nature parthenocarpy, but fruits can be obtained by treating the flowers with low concentration of oxygen. The flowers are treated with a low concentration of oxygen. See exact which part has been treated. In case of in the diagram A, you'll see the oxygen treatment in the to the flower, है ना. And there is a meiosis in the pollen development. In the pollen there is a meiosis and there will be a formation of pollen grains. So and this will leads to the it is the part of all, actually it is called as a parthenocarpy. It is also correlated with the apomixis also, because the self fusion is that. Apo means self, there is a mixis, means there will be no fertilization between the male and female gametophytes. So at that time only apomixis has been there, and yes, that apomixin is lead by the action of in case of parthenocarpic fruit by the action of oxygen, and but it is very low concentration. Parthenocarpy has also been induced successfully by synthetic oxygen in case of orange, banana, apple, tomato, pineapple. So these are the example where the parthenocarpic fruits are being synthesized by using synthetic oxygen. But in case of very little natural, that is apomixis part, apomixis it is also one correlated. We can correlate, not exactly studied, but it can correlated with the apomixis in case of formation of parthenocarpic fruit. So this is all about the oxygen discovery and the role of oxygen in the physiology. So the from the tryptophan, how the oxygen synthesized and what is exact role in the apical dominance, it is cell elongation, cell division, parthenocarpy, है ना, root development, root elongation. So these are the several applications of oxygen that we have studied today. So thank you very much. In the next lecture we will move towards another hormone, that is gibberellic acid. So thank you very much. We will stop here. In the next lecture we will see the different hormone. Thank you very much.